All right, chapter 15 is our special senses chapter. We're going to be working on the same sort of things we did in lab already, and that was looking over things like eyes and ears. But we're also going to add in the olfactory portion with the smell, as well as the equilibrium portion, which I know did work in with the ears and being able to see the vestibular cochlear nerve and seeing the vestibule area with the semicircular canal. So we'll just introduce those again real quick. And then the last one that we're going to look at is taste, so the gustatory portion. The eyes and the vision make up about 70% of the body's receptors, so thinking about how you are able to envision being able to identify things without seeing them. I think it's remarkable how a person who has a disability and vision impaired are able to have heightened or enlightened special senses such as taste and hearing and smell, because normal people, the normal population that has vision, relies on vision so heavily. So utilizing vision does make up a lot of our body's sensory receptors. There are some accessory structures with our eyes. We know that we have eyebrows, eyelids, but a couple of things that you may not have really paid much attention to are what's called conjunctiva and our lacrimal apparatus. Now, the conjunctiva, of course, is producing a mucus-like solution that goes over into the eyes and helps to cleanse the eye, and it's almost like an antibacterial material that comes through there. The lacrimal apparatus, of course, produces your tears, which help to cleanse the eye and move things through. And then the extrinsic eye muscles, which we discussed a little bit with our cranial nerves 3, 4, and 6, they have the ability to move the eyes, roll them around, push them up and down so that we can see more clearly. The eyeball structure itself, we remember there are 3 layers. There is the fibrous outer layer, the white layer, that's called the sclera. There is the vascular colored portion of the eye, that's the choroid layer. And then there is the inner portion of the eye, that's the retina. The internal eye is filled with a couple of humors, and we identified them as being the vitreous humor and the aqueous humor. The aqueous humor is a watery solution, and the vitreous humor, of course, is a gel. The eye does have 2 segments or chambers, if you want to call them that. There's an anterior and a posterior chamber. The anterior chamber is typically between the cornea, or the anterior clear structure on the eye, and the iris, and that is typically filled with the aqueous humor. The posterior chamber, of course, is from the iris all the way back to the optic nerve, or the retina portion, and that is filled with the vitreous humor. This is a beautiful picture of the eye from your lab or from your lecture book, and that way you can review the different parts of the eye. All right, I identified the portions of the eye already. There are 2 pieces of the fibrous layer. There is the sclera, which is the tough white outer portion, and there's also the cornea. And the cornea, of course, is the clear portion on the outside, but both of them do count as portions of the fibrous layer. You can see it's just a dense connective tissue. It's very tough. In the lab, we would do a dissection on this eyeball, and you would get to feel and see how dense and thick and tough it is. There's the cornea portion. It's about 1/6 of the fibrous layer, and it does allow light to pass into the eye. That way you can see. Without this clear cornea on the anterior portion of our eyeball, we would not be able to see anything because no light could come in. The vascular layer, as I said before, was mostly made of the choroid layer. It does include the ciliary bodies and the iris also. Those would be colored portions of the eye. And what we will find is that there's tons of blood vessels and lots of layers in here that are going to allow for nutrients and blood to get to each part of the eye. And that way it makes sure we have good vascularization. The ciliary body portion, of course, is just a bunch of muscles and capillaries that are secreting fluids that are helping hold things like our lenses in position and surrounding tissues of the lens and then controlling the shape. And then the iris, of course, is the colored portion on the anterior aspect that we see. If you look into your lab partner's eyes and you see those blues and browns and grays and hazels and whatever colors that we do have out there. That is the iris. And the iris is, of course, going to be the control factor of what is the hole in the middle that we call the pupil. Now, the pupil is nothing more than a central opening. So I always say this is a non-structure because it's a nothing. The pupil is just an empty hole. And a lot of people say, oh, my pupils were dilated. Well, really what happened was the iris was dilated and the iris changed the shape. And as the iris changes shape, it controls and lets more light in or it lets less light in depending on the size of the iris. As you can see, we have the iris in the middle here, and this is the colored portion. You can see it's two sets of smooth muscles that are going to allow for dilation or constriction. And if we have a parasympathetic response, if you remember back to chapter 14, it would constrict the pupil and help you focus on a small area. A sympathetic response is going to dilate the pupil, allowing more light in so that we can see a wider range area. The innermost layer of the eye is called the retina. It is a delicate tan-colored inner layer. It is pigmented on the outside, but this retina is extremely important because it allows for us to collect information. The retina has what are called the rods and cones. The rods and cones, and there's about 250 million receptors of these rods and cones, and this is what attracts or collects the light and sends the information to our brain. It actually sends it through this area called the optic disc. Now, the optic disc is referred to as a blind spot all the time because it's the one area of your eye that you cannot see. There's a neat lab protocol, lab exercise that you can do. And that lab exercise will allow for you to test for your blind spot. And the blind spot is where the optic nerve connects with the back of the eyeball. There are no photoreceptors there, therefore you can't see. Let's talk about the rods and the cones a little bit. First, the rods. The rods are there for your dim light perception, are also the most numerous. They do not allow any color images. All right, so think about this way. Our rods are there for kind of our dim, non-color, inordinate shapes. All right, so we can kind of see what's around us because of those rods there. But it's the cones that give our sharp, bright, high-resolution area. In the middle of these cones, you're going to find something called the fovea centralis, and it's a tiny area within an area called the macula lutea, and that is your absolute best vision. That's where you're most likely to see the most clearly. And a lot of people think that it's up close, but really your eyes are adjusted and they're shaped for a vision at 20 feet. So really they're adjusted for 20 feet. All right, so those are your photoreceptors for your eyes, the rods and the cones. Again, the cones are kind of like your HD vision, and your rods are there for like the Bobo standard division. So that you can, if we think about it like in TV settings. All right, the chambers, we said there's an anterior and a posterior, and I already mentioned before that the fluids are going to be mostly found in these chambers. The vitreous humor is the posterior segment, and you can see all the material that goes through there. It helps transmit and bend light. The anterior segment has the aqueous humor, and that is going to drain your the venous sinus, and it's going to have that watery solution that helps to bend light as it comes in also. Someone who have a blocked drainage there, you see that is what glaucoma is. So oftentimes we hear about older people with glaucoma, and typically that can compress the optic nerve and cause blindness. That is due to the glaucoma or blocking the drainage. Your lens is a biconcave or convex disc that's in there. It is very dense and thick. It's a connective tissue again, and it allows light to flow through. If we bend it or we expand it, it will allow us to see distances or up close, depending on what the shape is. Let's see. I don't think there's going to be anything in the test about wavelengths and colors, but just remember your eyes respond to visible lights, and that wavelength is in a color spectrum of 400 to 700 nms, and that is an electromagnetic spectrum. We think about that as being the color spectrum, and that is your Roy G. Biv, right? Your red, orange, yellow, green, blue, indigo, violet. That's where this color spectrum is. I posted this little thing about refraction on here because sometimes we get confused about what our fluid in our eye is actually doing. That fluid is creating a bending of the light, and that's necessary for us to send the signals into our brains the correct way. A good way for us to think about it is if we put a straw in a cup of water, you can see that the light bends when it hits the water, so that fluid creates a bend. It looks like the straw is broken or cut, but in actuality it's just the refraction or the bending of the light. Focusing the retina, pathways entering the eye. Remember, we have a specific pathways. Light has to go through our cornea, then through the aqueous humor. It then goes through the pupil and hits the lens. The lens will help bend the light. The vitreous humor will send it into the retina. The photoreceptors grab it, and that's what sends to our optic disc, which of course leads to our optic nerve. That will enter into the brain, and that is how we are going to collect light. As far as focusing for distant vision, like I said, our eyes are best suited for reading at 20 feet or looking at something for 20 feet. So that is going to be why we use the Snellen eye chart at a 20-foot distance. And that Snellen eye chart, again, was part of the lab for the eye section. We didn't get to do it in lab this time, but you will get a chance to do that at some point, I'm sure. Rods and cones, we talked about rods and cones a little bit already, so I'll kind of skip over these guys. But if someone is colorblind, they would lack their cone pigments, of course. They wouldn't be able to see the cones properly.